Hello everyone, and welcome back to more human physiology. So last time we talked about mainly the phospholipid bilayer. So we talked about the interesting case of the phospholipid in that it has a hydrophobic head and a hydrophilic tail, and how that really means that we need to construct our cell membrane out of a bilayer of phospholipids so that we can get hydrophilic interactions between the water in the extracellular and intracellular fluids and the heads, and then we also have the hydrophobic core so that everyone is happy and nobody is upset here. So in this particular video, we are going to start talking about membrane transport itself. But the first thing that we're going to talk about is the fact that the cell membrane is actually not exclusively made up of phospholipids. Now, the analogy I always like to use is that if you think of the plasma membrane as kind of like an ocean or a sea, the phospholipids are kind of like the water. Well, the ocean doesn't just contain water, so there are lots of different things that you can find in the ocean. Now, one of the main things that you're going to find in the cell membrane is a variety of different proteins. The proteins are kind of floating in the phospholipid sea, if you will permit that, kind of like icebergs. So the reason why these proteins are so very important is because depending on which types of proteins you find embedded within the cell membrane of a particular cell, those proteins are going to impart specialized functions to that particular cell. So these types of proteins that are found in the membrane, we call these membrane proteins. They come in two basic varieties. The first is called an integral membrane protein. Now, these are the type that basically go all the way through the membrane, meaning they are completely stuck in the entirety of the membrane. So this is going to include proteins like channel proteins, like what you see here. This looks kind of like a tunnel that goes all the way through the membrane. So you'll notice it goes from one side to the other. That's what makes it an integral membrane protein. Junction proteins that help to join cell membrane to cell membrane to give tissue stability and flexibility. Carrier proteins, which are kind of in the same category as the channel protein, as we will find out later on. And then many different types of receptor proteins, which we will talk about in week 3 when we start talking about communication. And then other than integral membrane proteins, you also have peripheral membrane proteins. Now, these are the type that are generally anchored to one side of the membrane or the other. So you're either going to find them on the intracellular side, like what you see here, or the extracellular side out here. You find them on one side or the other, but they do not go all the way through. Aside from proteins, which really proteins will be kind of the main focus of our discussion moving forward, you will also find carbohydrates embedded in the plasma membrane. Now, this is very interesting. So the carbohydrates can either be attached to the proteins found in the plasma membrane, or they can be attached to the phospholipids themselves. So if they are attached to the proteins, we call the whole thing a glycoprotein. And then if they are attached to the lipid, we appropriately call them glycolipids. So this is going to be very important in cell recognition as it pertains to things like the immune system, blood typing, the sorts of things that we'll talk about in greater detail later on in the semester. But something that you can go ahead and file away in your memory banks for now, something that actually is kind of important. Carbohydrate chains are only ever going to be found on the extracellular side of the plasma membrane, meaning they are always found facing out towards the extracellular fluid. You will never find them facing inward towards the intracellular fluid. So back to our major focus, which is membrane proteins. Receptor proteins, which again, we're going to talk about this a lot more when we start talking about cell-to-cell communication later on. Receptor proteins are a type of membrane protein that allows a cell to respond to a signal that originates somewhere else in the body. So receptor proteins are going to be basically how cells are capable of responding to input and output signals. So if you say have a control center that is responding to an input signal that comes from a sensor, Well, the input signal in some way is going to be some type of chemical messenger, whether it's a neurotransmitter that came from a nerve, whether it's a hormone that came from the sensor. That particular chemical messenger is going to have to bind to a protein on its target cell. And these receptor proteins are kind of like the antennae that allow these cells to receive and interpret those messages. So as I mentioned, these proteins, we're not really going to focus on them right this second, but they will for sure come into focus a lot more when we start discussing the nervous system, the endocrine system, and all the different ways that cells in the body can communicate with one another. So of all the membrane proteins that we are going to discuss in this chapter, our main focus we want to be on two of them, channel proteins and carrier proteins. Now, channel proteins are the variety like what you see here. It looks kind of like a hollow tunnel that kind of digs its way through the entirety of the plasma membrane. And really, that's not a terrible way to kind of think about it. So channel and carrier proteins are a little different, and we'll get into the difference later on, but they both basically accomplish the same thing. Both of them are going to be integral membrane proteins, and essentially what they are going to do is that they will permit a very specific type of molecule or ion to pass through the membrane under a certain set of conditions. Number one, that carrier or channel has to be in an open configuration. So as you can kind of imagine, if you look at the channel protein here, you can kind of see this hollow space that a molecule or ion could pass through. Well, that molecule or ion can't pass through if the channel is not open, meaning if there's a gate that is closed or something. So as long as the channel or carrier is open, it should be able to permit that molecule to go through. And then number two, the cell in question has to have or express that protein to begin with. So it is very possible, like say we have a glucose transporter, so a carrier protein that allows glucose to pass from the extracellular fluid to the intracellular fluid. Well, what we're going to find out is some cells in the body have that particular carrier, but many do not. So in talking about saying whether glucose could be permitted through or not, it's all a moot point if you're talking about a cell that doesn't have the channel to begin with. Another good example that we will see much later in the semester is the kidneys. Different parts of the kidneys express different types of ion channels and carriers, meaning certain parts of the kidneys can specialize in moving sodium and potassium. Certain parts can focus on moving glucose and amino acids, but very rarely are you going to see a cell that can move everything because it's got every type of channel or carrier. So hopefully what you can kind of appreciate right now, we're not really going to see kind of the big picture payoff for a little while, but hopefully what you can appreciate is that we don't want the plasma membrane to be this impenetrable barrier. So there are going to be circumstances in which different types of molecules and atoms have to be able to be transported across the plasma membrane, whether it's from ECF to ICF or ICF to ECF. So, for example, nutrients like glucose, fatty acids, amino acids, things that we get from the food that we eat, and then even oxygen from the air that we breathe, these sorts of things have to be imported from the extracellular fluid into the inside of the cell so that we can use cellular respiration to make energy for ourselves. If these things could not get through the plasma membrane, none of that would be possible, right? On the other hand, after cellular respiration is done, we are going to make waste products. We make carbon dioxide with every round of cellular respiration. We make urea by breaking down amino acids. So these are waste products that accumulate in the intracellular fluid, and we need to have a way of getting rid of them. So they need to go the opposite direction. They need to cross the plasma membrane to go into the extracellular fluid so that we can pass them out either through expiration and breathing or through urination or something else. And then also something that we will appreciate more when we start talking about the endocrine system, signaling molecules like hormones and neurotransmitters and things like that, they have to be secreted from where they are synthesized. So they have to be secreted, meaning that they are made in the intracellular fluid of the cell that made them. They have to be secreted out into the extracellular fluid. If we're talking about a hormone, that hormone is going to travel through the blood plasma, and then it will eventually get to the cell in which it needs to respond. And in some cases, that signaling molecule will need to go from the extracellular fluid into the intracellular fluid of the target cell. So there's kind of a lot that is going on there, but hopefully what you can agree on here is that it is a fundamental necessity that molecules in the correct circumstances have to be able to cross the plasma membrane. And then finally, the concentrations of things like water, the solvent, and then solutes like sodium and potassium and calcium and chloride and bicarbonate, and we could kind of list it off and on and on and on. These sorts of things have to be able to be maintained at a certain level, that's homeostasis again, for various reasons that we will probably get into once we start talking about things like the nervous system and the muscular system. Okay, so at this point, hopefully we understand, even if maybe we're missing kind of the big picture here, hopefully we understand that certain things need to be able to cross the plasma membrane. But as we said before in the first video of this chapter, the plasma membrane is what we call selectively permeable, meaning certain things can get in, but most things probably can't. So we need to, before we do anything else, we need to kind of lay down the ground rules that determines, okay, what is able to get through and what cannot. So our first clue in telling us what these rules are going to be is the actual chemical nature of the phospholipid bilayer. So that's really going to give us our first layer of control in determining what sorts of things can move and what sorts of things cannot. So here's a reminder of what a phospholipid bilayer looks like. You will recall that these little red heads here are the phosphatidylcholine heads. They are very polar and therefore are hydrophilic. And then the inner part of this, if you want to think of it as kind of the inner part of the sandwich, kind of the meat and cheese and lettuce and tomato of the sandwich, then that is a hydrophobic core. So we have hydrophilic on the outsides and then hydrophobic on the insides. So as we said, only solutes that meet certain criteria will be able to pass directly through the plasma membrane. So that's the other thing we want to make sure we are getting clear here. When we are talking about a molecule passing directly through the plasma membrane, we mean it is doing so without any sort of aid or help. It's doing so without the aid of a carrier protein or a channel protein or anything else. We literally mean it itself is squeezing in between phospholipids all by itself. So the membrane, being selectively permeable, is going to discriminate molecules on the basis of three qualities. So three qualities that the membrane will consider in determining whether it's going to let something pass directly through or not. The first one is probably pretty obvious. So we're talking about the size of the particle or molecule. So this one should be pretty intuitive. The idea here is that the larger the molecule is, the more difficulty it's going to have passing through the bilayer. On the other hand, the smaller a molecule or atom is, the easier of a time it's going to have getting through. So if we're only discriminating on the basis of size, then an atom would have a very easy time getting through, whereas a big molecule like a protein or a nucleic acid would have a very, very difficult time getting through. So if we go backwards here, and we look at these phospholipids again, keep in mind, the phospholipids are a little bit spaced apart, so there should be a fairly small space in between neighboring phospholipids that can accommodate a molecule trying to squeeze through. So obviously it should make sense that if you're dealing with a molecule that is very small, it should be able to kind of squeeze through one side and then kind of emerge on the other side. So the second issue is the polarity of the molecule. So the issue here is when we're considering the polarity of the molecule that's trying to pass through, we have to consider what that molecule would have to do in trying to pass directly through. So the main issue is that hydrophobic core. Any molecule that is going to pass from, say, the extracellular fluid through the hydrophobic core and into the intracellular fluid, that molecule is going to have to spend a lot of time interacting with those hydrophobic fatty acid tails. So therefore, those fatty acid tails are going to have a tendency to repel or exclude polar molecules. So the conclusion we can make here is that a polar molecule is going to have a very, very, very difficult time making it through the membrane directly, whereas a nonpolar molecule will have an easier time getting through because those nonpolar fatty acid tails are actually quite accommodating to fellow nonpolar entities. So far, we have talked about size and polarity. So as of right now, as of our discussion, the ideal molecule that gets through as easily as possible would be one that is very small and very nonpolar. The worst type of molecule that's going to have the hardest time getting through is one that is very large and is very polar. Now, there is a third thing for us to consider. Now, before we get there, the things to consider with these first two is that these are kind of operating on a sliding scale. It's not really a binary, yes, it gets through, no, it doesn't get through sort of thing. So the idea here is that, say, with molecular size, there are degrees of size, right? There's no such thing as like everything is either this small or everything is either this big. So there is kind of in-betweens there. So the smallest possible atom would have an easier time getting through. Then atoms get bigger and bigger, and then you get small molecules, medium-sized molecules, large molecules. So there's quite a few different things here. It kind of falls on a scale there, right? Same thing with polarity. There are scales to polarity. So it's possible to be totally nonpolar or totally polar, but there are kind of in-betweens there. So a partially polar molecule might have a little bit of difficulty getting through, but it might eventually get through. Well, the good news with this third quality is that this literally is an either-or proposition. Electrical charge. A molecule or ion is, a molecule or atom is either electrically charged or it is not. There is no in-between there. So if a molecule carries any type of electrical charge, it cannot pass through the membrane. There's just no way, no how. So the type of molecule we do want passing through is going to be one that is electrically neutral, meaning it carries no net electrical charge. So to summarize here, the type of molecule that can very easily pass through the membrane all by itself is going to be one that is very small, nonpolar, and carries no net electrical charge. A molecule will have a harder and harder time passing through if it fails any of these three, what you might call purity tests. It's going to completely fail if it carries any type of electrical charge, and it will experience greater and greater degrees of difficulty if it has any sort of polarity or is bigger than what you might expect. So this picture right here might kind of help us out a little bit. So we can actually see all of our properties and all of our qualities being put to the test here. Now, the easiest one that you can see right here is a very large macromolecule. So this is meant to be one of the globin proteins in hemoglobin that we talked about back in Chapter 2. This is a gigantic molecule relative to kind of the phospholipid. So it is so stinking big that it is going to have no chance of moving through the membrane. So you can see it is being completely repelled. A small nonpolar and non-charged molecule that you see right here is able to move through adjacent phospholipids to go from one side to the other. So that's a piece of cake. It's small, it's nonpolar, it doesn't carry an electrical charge, no problem. On the far right here, you can see electrically charged molecules like, say, sodium ions, potassium ions, and things like that. Because of their electrical charge, they cannot make it past those polar heads because, same thing with the polarity issue, the hydrophobic tails do not want to accommodate anything that has an electrical charge, and the polar heads will repel them as well. Now, the thing that is maybe going to be a little bit confusing here for you is you're probably focusing in on water here. So water passes two of the tests for sure. It is a very small molecule and it carries no net electrical charge. The problem and the thing that is a little bit misleading about this particular figure that I'm showing here is that water, as we discussed in chapter 2, is a very polar molecule, right? So why is it showing here that this gray arrow is indicating water moves directly through the membrane? Now, my argument would be that this figure is a little bit incorrect. So it is true that water can make it through the membrane to a certain extent, but the whole point here is that because of water's polarity, it's going to have a difficult time doing so. Now, we never said polarity is going to completely disqualify a molecule from getting through. We just said it's going to make it more difficult. And make it more difficult, indeed, it does. So water does have a very difficult time passing through the membrane all by itself. It does get through, it's just going to get through very slowly, which is something that we will clear up a little bit later. So now that we've had a chance to talk about the types of molecules that can and can't get through the plasma membrane, let's focus on the molecules that can, so things that are small, nonpolar, and not electrically charged. So we know that they can move through the membrane, but under what circumstances would they move across the membrane? Is it just whenever they feel like it? So we're in a science class, so there has to be kind of some order here. There has to be an explanation. Well, the direction in which molecules move, whether it's from ECF to ICF or ICF to ECF, this is determined by something called a concentration gradient, or the difference in concentration of a particular solute on either side of the membrane. So in this particular figure here, our solute is this small, nonpolar, not electrically charged molecule in blue here. And at the far left of this figure, meaning when we first begin, you will notice that all of these molecules are concentrated on one side of the membrane, and you find none of those molecules on the other. This is what we would call a very steep concentration gradient, meaning we've got a very large concentration of these molecules on one side and a very low concentration on the other. So the natural force that is going to move these molecules across the plasma membrane is they want to equalize the concentration. So over time, these solutes will naturally move from their area of high concentration to the area of lower concentration. But the important thing to remember here is that this movement from high to low concentration can only occur if those molecules can move. So if we were talking about a large polar electrically charged molecule, even if there's a concentration gradient, those things cannot move directly through the membrane, so they would be stuck on their side. So this first type of membrane transport that we are talking about is called passive transport. So passive transport is the process by which solutes are allowed to flow down their concentration gradients. So you can kind of think of this as, again, the same with energy that we discussed in chapter 1. It's a natural process that kind of tends to go downhill. So you can think of a high concentration as being kind of the high part of a ramp or hill, and then these molecules like to roll naturally downhill until they get to the bottom. So as long as a molecule is going downhill like this, this process of passive transport in which molecules move across the membrane from high to low concentration should not require any energy at all. So it would be similar to a catabolic reaction. So passive transport actually comes in two different varieties, and we categorize it based on the identity of the solute in question. Now, so far, we've talked pretty much exclusively about molecules that can pass through the membrane directly. So take an example like oxygen. A molecule of oxygen is a very small molecule. It carries no electrical charge, and it is completely nonpolar. So it is a perfect fit for the type of molecule that can move directly across the membrane all by itself. So anytime something like oxygen moves from high to low concentration across a membrane, we call this simple diffusion. So a good physiological example of simple diffusion, something that we will talk about late in the semester when we get into the respiratory system, is the movement of oxygen and carbon dioxide in the alveolar tissue of the lungs. So oxygen and carbon dioxide are both in the same category here. They are both small, nonpolar gases, and they can move down their respective concentration gradients by moving across membranes from high to low concentrations. So what you're looking here, this big pink sac right here, this is one of the inflatable sacs in your lungs called alveoli. When you take a deep breath in, your lungs are going to expand with air from the environment, and these sacs are going to fill up. This air that you have breathed in has a high concentration of oxygen and a low concentration of carbon dioxide. The blood that is moving into the pulmonary capillaries that are right in close contact with those alveoli, the blood that is coming in through the pulmonary artery is oxygen poor and carbon dioxide rich. So in blue here on the left side, we've got blood that has a low concentration of oxygen and a high concentration of carbon dioxide. Remember, in the air here, in the alveolus, it's the opposite. In the air here, it's oxygen high, carbon dioxide low. In the blood here, it's carbon dioxide high and oxygen low. So we've got concentration gradients for both carbon dioxide and oxygen. Oxygen will want to move from its high concentration in the air to the low concentration in the blood, and this is how, once the pulmonary vein takes this now oxygen-rich blood to the left side of the heart, to the left atrium, this is how we get our blood to become oxygen-rich. And then that high amount of carbon dioxide that is in the pulmonary artery blood, that is kind of a waste product that we want to expel out of the body. So we get rid of that carbon dioxide because it moves from high concentration in the blood to low concentration in the air, so that by the time we breathe out, we are breathing out a lot of that carbon dioxide that was originally in the blood. And we will talk about that much more when we start talking about the respiratory system. So the other type of passive transport other than simple diffusion is called facilitated diffusion. So facilitated diffusion is a term that is reserved for moving molecules from high to low concentration, just like what we did with simple diffusion, except this time we are talking about solutes that cannot pass through the membrane directly. So things that are either too polar or too electrically charged. Not too electrically charged at all, right? So some examples would be something like glucose, which is very polar, or sodium or potassium, which are electrically charged. Now, these are the sorts of things we said, okay, they cannot pass through the membrane directly, so they cannot and will not move by simple diffusion. But for both of these things, it is still possible to have a concentration gradient. We can relieve those concentration gradients by what is called facilitated diffusion. The trick here is that since these solutes cannot move across the membrane directly, they will require a channel or a carrier protein to allow them passage through the membrane. So if you think about how this kind of works, the reason something that is polar or electrically charged, the reason they don't like going through the membrane is because they have to interact with that hydrophobic core, and they don't want to do that, right? Well, a channel or a carrier protein basically forms a hollow tunnel through the membrane so that these things can pass through the membrane and they don't have to interact at all with the hydrophobic core, with the hydrophobic tails. Basically, the wall of this carrier or channel shields those solutes from the hydrophobic core. So the fatty acid tails don't know what's going on. The sodium or glucose or whatever don't know what's going on either. Everyone is kind of blissfully ignorant in that regard. But the important thing to remember here is that even though a channel or a carrier assists or allows the passage of things like sodium and glucose to go from one side of the membrane to the other, just because there is assistance happening here, that does not mean that energy is required. This is still a passive process because the whole thing is still going downhill. We are still going from high concentration to low concentration. Yeah, so facilitated diffusion is going to apply for both polar and electrically charged solutes. Now, you might be wondering, what about solutes that are really, really big? Well, it turns out that these channel and carrier proteins really are only going to be good for small molecules. So there's going to be kind of a separate mechanism for dealing with large molecules that we will cover a little bit later. So we are going to go ahead and wrap up this video right here. We still have one more special case of facilitated diffusion to occur, that being the case of water. But osmosis, the movement of water across a membrane by facilitated diffusion, is a topic in and of itself that is going to require quite a bit of time devotion on our part. So we are going to go ahead and cut this video off here, and we will start discussing osmosis when we come back. So thank you for your attention, and